Hi everybody. So I'll be giving a tutorial today on gene regulatory networks. So our group develops computational methods to look at gene regulation and gene regulatory networks. And I'm going to do a survey of different methods and touch upon some of the work that we've also done in the course of the different things. Because this is a pretty big topic, I'll be doing very superficial descriptions of methods, but if you're interested, I'm more than happy to provide you with details, and also I'll be at the picnic, so if you want to talk about stuff there. All right, so here's the plan for today. So we'll very briefly talk about just definitions of what are gene regulatory networks, but I feel like this audience knows what I mean by gene regulatory networks. And then I'll talk about methods, recent methods that have been developed to look at gene regulatory networks from bulk omic data sets. I just want to start my timer here. And then talk about some methods that are coming out for single cell omic data sets. So just so we are all on the same page, so what is a gene regulatory network? This is a network that specifies connections between regulatory proteins such as transcription factors, chromatin remodelers that control the context-specific gene expression level of a gene. And there are two components to gene regulatory networks. One is the structure of the network, which specifies which regulators define or control the expression of a gene. And the second one is the how. And methods differ in whether they are focusing on the structure or the structure and function, and how they model the function, and so on. So, that's what, and we depict these regulatory networks as these directed graphs. So why should we care about regulatory networks? Again, I don't think I have to tell this to this audience, but they are everywhere. They are important for a large number of processes. So for example, across evolution, we know changes at the gene regulation affects morphological complexity of species. In developmental processes, we know that there are transcriptional changes that happen that drive cell fate, and we know that disruptions in gene regulatory machinery can result in different diseases, and they can, as the speakers in the last two talks talked about, is they can be very useful for also interpreting genetic variation. But the challenge is that they are very difficult to define, and I'm mentioning two main reasons. One is that when a regulator controls the expression of a gene is determined by a bunch of different factors. So there are many levels of control. There are post-translational modifications, then there is long-range gene regulation, there is a chromatin state. And so that complicates our understanding of how and when a regulator regulates the gene expression of a gene. And the second is that these networks are fundamentally context specific. And context can be different things. It can be different cell types, it can be different individuals, tissues, conditions, and whatnot, and different diseases. And for most of them, we don't know what the gene regulatory network is. We don't know what are the regulators for the genes across these different contexts. And we are getting increasingly better in terms of mapping, coming up with better and better technologies to look at different components of the gene regulation machinery. This is somewhat of a dated slide from the ENCODE consortium, which looks at, which has used a large number of assays, and people in this audience have been involved in this consortium to look at what are the regulatory elements. So what I'll talk about today is methods that have been developed to try to leverage these types of data sets to try to tell us something about gene regulatory networks from a computational point of view. So, the first, or maybe the second part, is focusing on bulk omic data sets. So largely speaking, I would say there are two paradigms of thinking about methods that have done gene regulatory network inference. And I've called them I've put them in these names. They may not be the best way to name them. If somebody has a better way to name these methods, I'm all ears. But so one class of methods is what I call expression-based network inference methods. And I'll tell you about that. But the general idea of these methods is that they think about gene expression as the primary source of data they want to describe. and auxiliary data sets are used as trying to inform the structure of the model that they are inferring. And the second class of models are in the family of what are called predictive models of gene expression. And these models are incorporating a lot of information that is on the promoters or enhancers of genes and are trying to predict gene expression as a function of upstream regulatory machinery. So let's talk about the first class of methods, and these are expression-based network inference methods. And there are, so this is the vanilla type of like method that what you would do. And what these methods do is they start with a large number of gene expression matrices or gene expression measurements and use different modeling formalisms and outcomes a network that connects regulators to target genes. And so there are a lot of methods out there, but the general principle of all of these methods is they are really, so the regulators of a gene are really those that explain the expression of a gene, of a target gene well. So they are trying to learn these predictive functions. So the psi can take the form of a Boolean function, it can be a probabilistic function, it can be a differential equation, and all of these methods are really trying to predict or figure out what should be the regulators, that what are the covariates that explain the expression of a gene. So the regulators of X4 are those that best predict the expression of a gene. And this is a not exhaustive list of methods that are out there. They vary depending upon whether they are using a more probabilistic graphical model type of framework or an information theoretic type of a framework. Are they learning pairwise relationships? Are they learning higher order relationships and whatnot? And so this field has been there for more than 20 years now. So I thought maybe I'll say a little bit about how the field actually has evolved over the last 20 years or so. So in 2000 was when these methods were, Friedman and Aviv Regev and Daphne Collar and Dan Appel, this group of people, they formulated probabilistic graphical models or Bayesian networks to formalize what a gene regulatory network could be, could look like mathematically, and they came up with the first set of algorithms to infer them from gene expression data. And then there were information theoretic methods, methods to model perturbations. And then in about 10 years ago, people realized that it's not sufficient to learn a single model. Let's try to do some subsampling or bootstrap, and that really helped to try to improve the quality of the networks that we get. And in particular, this method called Genie3 has been used also with single cell data and has had a lot of mileage. And then the other methods then try to incorporate cell type specificity or incorporating constraints in the model. And then in the last few years, most of the methodological development has been focusing on single cell, which I'll talk about towards the last, in the third part of my presentation. In the general area of bulk omic data types, I would say for integrative expression-based network inference, methods can be grouped into two main categories. So what these methods are really trying to do is, in addition to gene expression, can you see my pointer? Okay. In addition to gene expression, they use these additional sources of data such as accessibility or histone modifications to inform the structure of the network, and they do this by putting in a prior. And so you can use that to guide the structure of the network. And the other thing that people have done is to relax the assumption that mRNA of a regulator should be used to predict the mRNA of a target, and they use something called transcription factor activity estimation. So I'll touch upon briefly these methods. So the first is prior-based approaches for network inference. So what these methods try to do is, given gene expression data and auxiliary data sets such as accessibility, what we want to do is predict the regulators of a gene based on its gene expression levels, but don't only use gene expression, use these other sources of data to further guide the network inference approach. And methods here can be grouped into two classes, and one class of methods which are parameter prior based methods are basically coming from those approaches that have used something like a linear regression type of a model. And so there, the predictive function that relates the regulators of a target gene are all linear functions. They use regularized linear regression, and they put in this prior knowledge inside the parametric form of the model. And then there are other methods that are saying, let's not marry ourselves to how the regulators will relate to the level of the target. Rather, let's try to impose this information to guide the structure of the network. So in the first class of methods, one method that has been quite popular is Inferelator, and it's actually a suite of methods. They have a large number of methods. They also have single cell methods. And this method, it can be used for time course as well as single time point data. And the general principle of this method is that it makes use of regularized linear regression. And so what this is saying is, let's try to learn a sparse model. Let's try to use the smallest number of regulators to explain the expression level of a gene. And the way this is done is by putting in a penalty. So you pay a price when you're adding a regulator in the model. But then if you have prior information, you pay less penalty. So that's the idea. So just to tell you a little bit more about that. So this is one of their implementations. This is called modified elastic net. This is the objective. This is the overall error. So yi, so I is the gene whose expression we are trying to explain. This is the overall error that you are trying to minimize, and these are the different regularization terms. And so this is an elastic net penalty. And what they're saying is, let's put in this additional term theta IP over here, which can be used to tune how much penalty we want to put on the regression weights. And so if I have some prior knowledge of a particular regulator P, I will add less penalty. So this theta IP is less than 1, so I will pay less cost. So that's one way in which you could do this. What we've done is use something based on a structure prior based framework. And so here what we've done is we think of the thing that we want to infer, the graph G, as a high dimensional random variable, and we want to put priors on the graph structure. And that prior is informed by these different auxiliary sources of data. And what we are saying is that, so we want to put a prior probability of an edge based on these different data sets, and then when we see a gene expression data, then we will have a different, what we'll get is something that is informed by both the prior as well as the data. And we've done benchmarking studies using yeast as well as mammalian systems. And here is, so what we've done is taken gene expression, used a motif-based prior, inferred a network, and then asked whether adding priors actually helps. And this is a AUPR curve, which shows that so there are two clusters of methods here, methods that use a prior and methods that don't use a prior. And we can see that methods that use a prior are generally much more beneficial in terms of inferring the graph structure. Okay, so the other direction where methods, yeah, question?
What do you mean by motif-based prior?
Okay, so what I mean is, so transcription factors have sequence-specific motifs.
Yeah.
And so what you can do is you can basically scan the promoters of genes, and then you can say that this gene has a significant binding site for this transcription factor. And so that can, so you can use that as a way to say, okay, this transcription factor likely regulates the gene. So that's what I mean by motif-based prior.
But you're not actually looking at data sequences?
Not yet, not yet. Yes, question.
Can you say a bit about what data is used to evaluate this and where the truth annotations for the...
Yeah, so the thing is, I will actually not talk much about validation of gene regulatory networks. That's again, so the ground truth, so the problem with GRNs is that there's not very much ground truth. So what you would do is, So one way to deal with it is, so there are like things like yeast and human, you have in human embryonic stem cells, for example, you have some databases that have collected TF target connections. So by doing regulator knockout experiments and doing genome-wide gene expression measurements. And so you could use that as a way to validate, to serve as a ground truth. In yeast, also there are lots and lots of like ChIP-seq experiments that have been done. So those would be considered as experimentally derived ground truths that you could use to validate your gene regulatory network. And for the yeast, in this case, we were using. This was based on a evolutionary conserved ChIP experiment to look at the quality of the networks. But we also have like knockout based validation data sets. Does that answer your question?
Thank you.
Okay, so more questions? No. Okay. So, all right. So the second direction has been what is called estimating transcription factor activity. So one of the big criticisms with using mRNA level of a regulator is that we know that it's not really the mRNA level of the regulator. It's actually the activity level, that it's maybe the protein level is much more closer to what we should be using in our network inference algorithms. But we typically don't have protein levels. So can we actually relax this assumption where we're using the mRNA level of a regulator to predict the expression level of a target gene? So the idea is called transcription factor activity estimation. And so in this approach, what is done is we assume that the gene expression matrix that we have is a function, is a product of two matrices A and P. And so I'm going to first tell you about how to estimate the activity level of a transcription factor, and you will see that it needs a network. And then we will use that activity to again infer another network. So it needs some partial regulatory network that may or may not be correct, but it's going to use that to estimate the activity level, and then we are going to use that to infer the gene regulatory network. So TFA is estimated using something called network component analysis. And this was actually formulated several years ago. And so the general idea is, so you start with this A matrix, and this is informed by by an input network. And then you basically iteratively estimate, given a A, estimate the P, given a P, estimate the A, and you keep doing this until you're done. And then once you're done, this P matrix is what gives you the activity of the regulators. And once you have those things estimated, then what you can do is use the estimated activity levels and the gene expression levels, and then infer a gene regulatory network where a transcription factor can come into play based on its mRNA levels as well as its transcription factor activity levels. Now, the question is, how do you create the input network, and then how do you know how much does it actually matter? If you, for example, if you had a very good input network from the get-go, do we really need to do gene regulatory network inference, right? We've kind of solved the problem. So, so one way is, so this kind of maybe answers Smita's question. So to create the input network, you can use sequence-specific motifs, possibly refined by accessibility, and you can use that to get the first initial network. But the second thing is the quality of the network can have an effect. So, so we did a simulation where we considered different sources of different extents of noise in the network. So we added a bunch of false edges, and we asked if we just did this standard approach of TFA estimation, what happens? And we see that in simulations, the performance, the estimation of TFA actually becomes worse and worse. So this is correlation between the true TFA and the estimated TFA, and also the quality of the networks that you get are also getting worse. So what we did was use a regularized version of TFA where we said for most of the situations, the network that is used to estimate the activity has some notion of confidence. And so we could use that to constrain how we are actually estimating the activities and also the network structure. And basically, this can be formulated within an L1 kind of a penalty, and then it can help to do network inference. So again, based on simulations, what we found is, so what we call our method is NCA plus lasso, and based on correlation and AUPRs on simulated data, we can see that the quality of the TFAs that we get with regularized TFA is better. And so the output of a method is a network, but also a TFA, and you can use that TFA into other algorithms that just do regression or also have the ability to incorporate priors. And these are different methods. MERLIN is our method, but these are all the other methods. And we see that using regularized TFA is definitely beneficial compared to not using regularization. So with that, I just want to summarize. There are different methods out there to try to incorporate gene expression and use that together with other sources of knowledge. But the general idea of these methods is we are trying to explain gene expression levels, and the thing that we're trying to explain it with is this gene regulatory network. But we know using gene expression is not sufficient. Can we use these other sources of knowledge to try to constrain the regulatory structure, the structure of the gene regulatory network? Okay, but the important thing to keep in mind is gene expression is the primary source of data that we are trying to explain. Okay, so now is the other class of methods, which are based on, for the lack of a better term, I call them predictive models of gene expression. But these methods are trying to predict gene expression and based on other features, but their goal is not to actually predict gene expression. The goal is to actually try to find what explains gene expression, what are the key regulators. And so here's an overall very basic schematic of these approaches. We start with a set of genes that might be interesting, and by interesting it could be genes that are differentially expressed, highly expressed genes. And then we want to predict their gene expression level or gene expression state as a function of their cis regulatory elements. Okay? And once you've learned this predictive model, to understand what actually drives gene expression, we would look at the model, what helps the predictions, look at the model coefficients, and then figure out what are the important predictors. And there have been several methods out there, and they vary depending upon the type of function they use to try to link upstream regulatory machinery to gene expression. So the first set of methods, and still used, are linear regression based methods. So here we have a vector y, and we have features that are really promoter features like transcription factor binding signals or histone modifications, and we are trying to estimate what is this expression level as a function of these different features, and we fit a linear function. Then there were tree-based models from Anshul's group, Anshul Kundaje, where they try to capture more non-linearities. And then more recently, people are using deep learning-based methods to try to connect regulatory features to gene expression levels or gene expression states. So like the expression-based network inference methods, these methods have also evolved, and they also started around 2001 with the reduced method from Herman Bassumaker's group et al., where they tried to predict gene expression as a function of sequence features or sequence motifs. And then methods that were trying to either increase the complexity of the model, like tree-based methods, incorporate temporal dependencies, including methods from Jason's group, DREM, and then so Gonzalez et al., this is a paper from Christina Leslie's group. There they try to incorporate accessibility to inform these predictive models. And more recently, basically we are seeing methods that are based on deep learning based methods. In particular, this method, GraphReg, tries to combine convolutional neural nets and graph neural nets, and that helps to actually bring in long-range regulatory programs like enhancers to predict the gene expression level of a gene rather than just focusing on the promoter of a gene. So, so what do these models actually do? So I just want to give a snippet of the different types of outputs that you can get from these models. So this was a paper from, again, Christina Leslie's group, where they looked at 6 different cell types. These are associated with hematopoiesis, and they measured RNA-seq, different histone modifications, and this is a particular locus where you can see these different assays. And what they did was they identified differentially expressed genes between B cell and HPSC, NK cell and HPSC. So they identified what genes are differentially expressed. And then for each of the genes, they had features based on the accessibility, motif features, and the histone modifications. And then they learned linear regression models to explain the expression level of these genes. And what you're seeing here is the regression weights that these models actually learn. Actually, regression weights times the features associated with a gene. And these are one set of genes, and this is another set of genes. And you can see that there are some regulators that are specifically associated with certain sets of genes. And so, for example, the P1 regulator is associated with the B cells. So that's nice, because what you can now identify using these models is what are the key regulators that explain the expression of this set of genes, the high genes versus low genes, and so on and so forth. Something that we've done is these methods have taken one cell type at a time. So I have a gene expression level, gene expression states. Can I explain the gene expression levels of this cell type? But many times you have a time series or a lineage kind of a thing where you have multiple cell types and you have some relationships across these cell types, and you have these different types of omic data types. Can we use that to infer a gene regulatory network? And we developed a method called DRMN, where we leverage the lineage information, sequence-specific motifs, and then we use this information to partition genes into high, medium, different gene expression levels, and then we also predict what actually drives those gene expression states. And we've applied this to a bunch of different data sets. I'll talk briefly about a data set that we used, again, from Jason and Catherine Platt's group from UCLA. This is a very nice data set, and one of my collaborators also studies cellular reprogramming, so it kind of makes sense. That's why I looked at this. So this is a linear time course where you go from a differentiated cell type, which are MEF, mouse embryonic fibroblasts, and you're going to the pluripotent state captured by the embryonic stem cells. And they've measured a whole bunch of different types of data sets. So they've measured histone modifications, transcription factors, and also gene expression. But keep in mind that these data sets as such cannot be used for methods like expression-based network inference, because there's only one sample or maybe 4 gene expression levels. So you can't really do regression here for individual genes. But what you could do is you could group genes into sets and then try to explain the gene expression level of sets of genes based on the features. So, here is, so as I said, DRMN enables us to kind of identify gene expression, groups genes into different gene expression states. And so these are different sets of states. So there were 7 states, with red representing high expression and blue representing low expression. And then these are different regulators that we predict. We have histone modifications as well as transcription factors. And we can also look at how individual genes change their expression state on the lineage or over time, and then we can ask what actually explains those expression levels of those genes, and we can predict regulators like transcription factors or histone marks. All right, so just to summarize what I talked about in terms of bulk omic data sets, there are 2 main ways in which you can use data to try to multi-omic data to infer gene regulatory networks. One is expression-based network inference, where you are trying to predict expression as a function of expression of the regulators. These methods need a large number of measurements to be able to solve these regression problems, or linear, nonlinear regression problems. But the advantage of these methods is that they can use different types of regulators. They can use transcription factors, signaling proteins, or different kinds of regulators into transcription factors that may or may not have motifs. And the way the non-expression data is being used is as priors in the model to inform the graph structure. On the other side are models that are also integrating, and they are much more integrative in a sense, because all the other omic data types are really informing the gene expression in a more direct way than the expression-based GRN inference methods. But you can only use transcription factors as potential regulators, and also transcription factors for which you know what the motifs are. Expression is predicted from the cis regulatory features. But the advantage is that you can work with very small samples. So because they are really trying to work at for a set of genes, what are its features, can we explain the expression level of these genes. All right, so that concludes the bulk part of my talk. In the rest 10 minutes or so, I'll talk about what has happened with single cell genomics and what methods are out there for inferring regulatory networks from single cell omic data sets. So I guess this is an outdated slide really. Right now, single cell genomics is really transforming biomedical science. And there are many different individual labs as well as consortia efforts, and they're just generating lots and lots and lots of data that are out there. And this has also been very transforming for the gene regulatory network field, because now the sample size issue has gone away. Now we have lots of measurements, and there have been lots of methods to infer gene regulatory networks from single cell omic data sets. So what's the general idea? We have genes along the rows, and then we have cells, individual cells along the columns, and you feed that into a network inference algorithm, and out comes regulatory connections between regulators and targets. I should say connections between regulators and targets. These are all statistical correlations. And there have been several methods out there, and you can check out these good reviews that talk about methods for inferring gene regulatory networks from single-cell RNA-seq data. And there have also been benchmarking studies which have tried to compare different single-cell RNA-seq network inference methods. I'll briefly touch upon them. So in terms of the class of methods that are out there, we can group them based on how they model the relationship between a regulator and a target gene. Again, you could use information theoretic methods, and there were some new methods like PIDC that and Kane and Remy, for example, from Smita's group, that have used information theory as a way to relate regulators to target genes. There have been graphical model-based methods, simple correlation-based methods, and these methods basically differ in really how do we link. How do we explain expression level of a gene? What is the mathematical form? What type of function should we use? And then with single cell data, you can also say another thing that people have done is order cells on a trajectory or pseudotime. And there have been methods that have tried to leverage pseudotime to inform the topology of the network. It's still debated whether it's actually useful, considering that pseudotime in itself can be noisy. Depending upon how you estimate pseudotime, you may get a different network, a different pseudotime or ordering. But there are methods out there that have incorporated pseudotime, and methods that actually don't incorporate pseudotime. And very recently, methods are actually going away from just using single-cell RNA-seq, but rather leveraging other omic data types, in particular accessibility. And here are a few methods that are out there, including one from our group, and I just wanted to briefly talk about those methods. So let's say that you had accessibility information for individual cell types. So here I have two cell types, C1 and C2. What you could use accessibility as a way to get a graph structure. So for example, you have two cell types and two genes, G1 and G2. And in one cell type, this gene G1 promoter is accessible for this transcription factor, whereas it is not accessible in this cell type. And accordingly, the other gene has an accessible promoter, whereas it is not in the other cell type. And both of these would give you different cell type specific networks. And so with accessibility, you could actually map a skeleton gene regulatory network that is cell type specific, if you had cell type specific accessibility information. And there have been methods that use this structure as a given and then add gene expression on top of it, but don't change the structure of the network, rather subselect out edges to inform, to learn a gene regulatory network. And then there are other methods that actually use accessibility derived gene regulatory network just as a prior. And the reason to do that is, again, these methods would rely on the sequence specific motifs of transcription factors. So to be able to incorporate transcription factors that don't have motifs, we need to account for that. So it's better to use the accessibility information as priors and then use gene expression as an additional source of data to infer gene regulatory networks. So I'll talk about really very briefly about three methods. The one method is Symphony, and this is from Dana Pear's group. And so this method takes in bulk attack-seq data and single-cell RNA-seq data, and it is able to deconvolve the attack-seq data into cell-type specific accessibility, and then use that deconvolved cell-type specific accessibility as priors to in the graph structure. And the graph is being modeled as a Gaussian graphical model, and so it solves the problem of deconvolution and inferring a gene regulatory network. So in a way, it is defining the cell types using the gene regulatory network structure. There is a lot of like heavy duty inference under the wraps. If you're interested, please check it out. And they've used it to look at some single cell cancer data, and they can find key regulators. And one of the points that they made is some of the regulators that they identified were actually did not have a known motif, and therefore incorporating gene expression was really important. So that's the Symphony method, so bulk attack. Then there is a suite of methods from the Inferelator 3.0 package. It's a nice package. It's a collection of different network inference algorithms from Richard Boneo's group. The one that was specifically made for single-cell RNA-seq is called AMUSR. And so this method, it takes in single-cell RNA-seq cell clusters. So you have to define those cell clusters, and for every cell cluster, you have to define a prior network. And this can be the same for all the cell clusters, or it can be something that you may have, accessibility information. You can use that to define a cell-type specific prior network. And the output is a GRN per cell cluster. But what it does is, instead of learning the gene regulatory network for each cell cluster individually, which you can do, they instead say, let's think of the network inference problem for multiple cell types as a multitask learning framework, learning problem. So each task is a network inference problem. Let's try to solve this jointly, and that's what they do, and it works quite well. Something that we've done is take this multitask learning framework, but realize that cell types themselves can have more structured information. So they can be related by a tree, a cell lineage tree. And so we assume that we have RNA-seq data for individual cell clusters and also accessibility information. And then we feed that inside SEMTNI, our algorithm, and then outcomes for every cell cluster, a GRN. And we just got the preprint out, so if you're interested, please check it out. So here's a simple workflow. So we've applied this framework to cellular reprogramming, where we have already an attack and also a published data set for hematopoiesis. We used the Liger method to integrate single-cell RNA-seq and attack-seq. And the output is, for every cell cluster, we have RNA and attack matched up. Then once we have cell clusters, we used a simple minimum spanning tree approach. We applied different algorithms. And then once we had the networks, we actually did some dynamic network analysis. Just to give you a sense, so these are the Liger clusters, these are the Liger clusters and the different samples. And this is what the cell lineage tree looks like, where C4 is largely the MEF cells, and then C5 is largely the ES cells. And then these are different cells that actually don't reprogram. And so there are really these two branches. So one interesting question is, why do these cells actually don't reprogram? And so we've tried to answer these questions by looking at the networks, and this is just a snippet. This is basically each of these little blue-red pictures are really GRNs that we identify in each of these different cell clusters. And then what we see is, if you look at the topology of the network, you can see that there are some regulators like ESRRB that become more and more prominent that were not there in the beginning. which are actually not there in this particular lineage, which can be used as a way to prioritize. So regulators like WT1, which tend to just stick around, may be potential reasons why maybe these cells don't reprogram. All right, I'm really done. I'll just summarize. In terms of gene regulatory network inference, we can use bulk data, and recent methods we can group them into those that are using other omic data types to guide an expression-based network inference algorithm, or try to think about predicting expression as a function of these different cis regulatory features. And from single cell omic data sets, there's a lot of opportunity, and the vast majority of the methods you just use single cell RNA-seq measurements, but more recent methods are trying to use attack-seq and also model the fact that cell data sets that we're getting really have a bunch of different individual populations, so we need to define those populations and infer population-specific gene regulatory networks. Some things that we didn't talk about but very important is validation of gene regulatory networks. How do we do it in a systematic, high throughput way? Causal networks, and now with perturb-seq data available, maybe we can infer gene regulatory networks, but maybe Harold has other thoughts. I could try to find him. And then some things that also that I didn't talk about is annotation of regulatory elements. These are key components of gene regulatory networks, and then long-range gene regulation. And with that, I just want to thank my group and different funding agencies, and I can take questions.